Okay, so different patterns of development in the animal kingdom is going to be the last topic for chapter 27. Remember that we talked about how a zygote divides to form an embryo called the morula. So like the 2-cell, the 4-cell, the 8-cell, the 16-cell embryo is the morula. And then somewhere around 32, 64 cells, something around there, the embryo hollows out. The cells move to the surface. They keep dividing, but the center of the embryo itself is hollow or full of liquid. And that's called the blastula. And then the blastula, the cells keep dividing, and on one particular point on the embryo, the cells start migrating inward into the space inside the embryo, and that process is called gastrulation. Now, gastrulation is the point at which the embryo now has 2 germ layers, and it has an opening to the outside we call the blastopore, and the blastopore eventually is going to be an opening to the digestive system. And then the inner part of the embryo is called the archenteron, and that's the primitive digestive chamber. So let me make a really crummy drawing for you. So the embryo would have the outer layer of cells, and then the cells that migrated inward, I guess, looks a little bit like this. And this opening right here, where this dot is the blastopore. So the blastopore is actually just an empty space. It's an opening. And then the digestive chamber is behind the blastopore. It would be right here, and that's going to be the digestive cavity as the embryo continues to develop. Now, this space inside here is called the blastocele. Blastocele. And the root cele refers to a cavity, a space. So now you have the digestive space, which has an opening to the external environment, and you have the blastocele, which is not directly connected to the outside environment. It's completely enclosed. Okay, so there are 2 ways that development can happen, and really we're starting from the gastrula stage at this point. So we're about to go past the gastrula stage in our discussion, which we haven't done before. And all of the organisms that can do this, one of these 2 developmental paths, are bilateral and they're triploblastic. So on that branch of the evolutionary tree, you then have a split, and some of the animals follow a series of events that cause them to be classified as the protostomes, and some of the animals fall into a different branch called the Deuterostomes. Stome refers to opening or mouth. So you've probably seen, no, I don't want that pointer or pen. So stome refers to an opening, in this case, mouth. So you'll see that in some cases. Protostome means the mouth is the first opening. Proto referring to sort of the beginning, the first. Deutero refers to the second. So in the Deuterostomes, the mouth is the second opening. which is kind of interesting. All right. So there's a few issues a few characteristics that go with the protostomes, and then a few characteristics that go with the deuterostomes. So you just learn collectively all the characteristics of the protostomes and then collectively all the characteristics of the deuterostomes and keep them separated. So maybe a chart in your notes would be helpful. The first thing is what we call the fate of the blastopore. The fate of the blastopore, remember, is the opening to the archenteron, which would be right here. So this would be the blastopore. And historically, we believed that protostomes, the blastopore became the mouth. So you can see the way this embryo is oriented here. The blastopore, later on, it's the mouth. In the deuterostomes, the blastopore, if you keep the orientation the way it is in this picture, you can see that later in the animal, that's the anus. So if the blastopore forms the anus, that means the anus is the first opening, and then the mouth is the second opening. That's why it's called the deuterostomes. Now, there is some, we're not sure now that this is actually correct, but that's why it's called deuterostome, is that it means mouth second. So in any case, the differences between these two have to do with how the mesoderm forms. In the protostomes, the mesoderm forms as a solid block of tissue right in the blastocele, but near to the side of the embryo where the blastopore is. And then it splits and sticks to the ectoderm, which is shown in sort of a blue-gray here, and in this picture the mesoderm is shown in red. So after the block of mesoderm splits and sticks all the way around, you end up with a coelom that is completely surrounded by mesoderm. And this is another coelom completely surrounded by mesoderm. So that's the body cavity. Now remember, you have another cavity in the animal that's called the digestive cavity, but we're talking about the coelom. In the deuterostomes, the mesoderm forms directly from the endoderm, and the endoderm is in this inner part of the gastrula embryo, and then the mesoderm branches off, or we say pinches off of the endoderm. So what we have here is in the protostomes, a block of mesoderm splits and lines the coelom, and that splitting process is called schizocele. You may know schizo means split. So the coelom forms by the splitting of a solid block of mesoderm, and it splits, it sort of unzips and creates an empty space in the middle. The deuterostomes have mesoderm that comes off of what's going to be the digestive cavity. So that process is called enterocele, because the term enteric means having to do with the digestive tract. And so the coelom is lined by mesoderm that buds from the digestive tract, enterocele. So enterocele is a process that is connected to the deuterostomes. Schizocele would be characteristic of a protostome. So let's say you had an animal embryo and you didn't know what it was. If you could watch it, if you had a really good microscope and you could watch it, and you were somehow able to visualize the mesoderm, then you would at least be able to classify it as either a protostome or a deuterostome based on how the mesoderm forms. And in the end, though, the thing about it is when you if you don't watch the embryo very early, if you just look at the end product, you really can't tell, because both of them in the end look virtually the same, just in terms of the way the structure of the coelom is. So you would have to watch them early and watch the coelom forming. Okay, so formation of the mesoderm is one of the features that differentiates protostomes from deuterostomes. So protostomes have schizocele, deuterostomes have enterocele. So that's the first thing. Number 2, cleavage pattern of the embryonic cells. I'm going to launch this video here, because in this case an animation is worth 1,000 words. But this cleavage pattern means what happens when the 4-cell embryo becomes the 8-cell embryo. At least that's the easiest time when you can view this. So you have a 4-cell embryo, and then each of the cells divide, and you end up with an 8-cell embryo. But the way it looks is different in the protostomes versus the deuterostomes. In the protostomes, it's called spiral cleavage. In the deuterostomes, it's called radial cleavage. But you really have to look at the videos. So I'm going to play these for you. Okay, that's the 2-cell embryo, the 4-cell embryo, and then the 8-cell embryo. Let's do it again. 2-cell embryo, 4-cell embryo, 8-cell embryo. Do you see how when you go from 4 cells to 8 cells, the top 4 cells spin out? All right. They sort of spin. Let me get back to this. Now let's compare that to the deuterostomes. The deuterostomes, it's called radial cleavage. So let's watch this one. 2-cell embryo, 4-cell embryo, 8-cell embryo. See how it stacks directly on top? From 4-cell to 8-cell, it doesn't spin, it just stacks up directly on top. So you might want to watch those again. All right, so spiral cleavage versus radial cleavage. And then the developmental fate of cells. Protostomes, as the embryo divides, as the cells divide in the embryo, those cells from a very early point in time already know what their final cell type will be. I'm not saying it's from the first cell, but from when you're, at 8 cells, 16 cells, the cells already start to be limited into what they're going to become. And we call that determinate development. It's a much more rigid embryonic development. And if you take an embryo that's, 64 cells, and it's a protostome, and you take a laser and you kill one cell with the laser so that you have 63 cells, what will happen is those 63 cells will continue to divide and try to become what they were supposed to become. And you could end up with an animal that is missing some part in the end, because the one cell you killed is was supposed to become a certain structure, and then now it's just not there. Sometimes that means the animal dies. And that's in contrast to the deuterostomes. The deuterostomes, the cells do eventually determine a certain type, but it comes much later. So the early embryo, the cells can adjust. So if you take a 64-cell embryo from a deuterostome and you use a laser to kill one of the cells, the cell next to it will sense the missing cell and just divide again to replace it. Because in that embryo, they're stem cells. They're just all the same. So in deuterostomes, indeterminate development means the cells are stem cells much longer, and stem cells can become anything. And so there's a much more flexible developmental pathway with the deuterostomes, because the cells stay in that stem state much longer as they continue to divide. It's not that one's better than the other. It's just two different ways of development. It also means that deuterostomes can have twins, because if you have a two-cell embryo and the two cells get separated, each cell will readjust and just form an entire organism, and you'll get identical twins. If you have a protostome and the two-cell embryo separates, you'll just get like the front part of the animal and then separately the back part of the animal, I guess. And you won't get twins. And actually, they'll probably just die. But you can't get twins in the protostomes because the cells don't function like they do in the deuterostomes. So deuterostomes, you can have twins. All right, so this slide just reviews the different issues, the different parts of considerations of symmetry, tissues, body cavity, patterns of development that we've gone through. And I'm going to stop this segment here, and then we'll do one last segment that looks at deep time a little bit in the animal kingdom.